Welcome back to my channel. It's Doc Janelle. The subject at hand is still Human Anatomy and Physiology. And this is the second part of our Cell Anatomy and Physiology. In the first part of the video, we talked about the cell membrane and the different transportation methods that substances and water use in order to traverse into the cell. And for the second part of the video, we'll be talking about cell anatomy and the different parts of the cell, the different organelles, and the functions of each and every part. So, of course, as mentioned, organelles are the specialized structures that perform certain functions inside the cell, and they are all floating in a jelly-like substance called the cytoplasm. Let's first review the appearance of the cell in general. The blue part over here that you see in the most exterior part is the cell membrane. The yellow substance that you see all over the cell is the cytoplasm. In these different structures and shapes, that colors that you see are the different organelles. So as mentioned, the cytoplasm is located inside the cell. It's characterized as a jelly-like fluid, and it comes in contact with the inner leaflet of the cell membrane. And the main function really of the cytoplasm is that it holds the organelles in place. Okay, this is where the organelles float. The next part is the cell nucleus, and out of all the organelles, the cell nucleus would probably be the most prominent. By most prominent, what I mean is, siya yung kitang-kita talaga if we look at a cell under the microscope. You will not miss the nucleus. It's usually located at the center of the cell, as you can see here in these diagrams. And all cells in the body would usually contain a nucleus, no? But there are some cells that would lose their nucleus when they become matured. And one example of that is your red blood cell. But then again, at one point in its development, the red blood cell had a nucleus but just lost it near its maturity. Okay? Now, what is the function of this prominent organelle? The main function of the cell nucleus is that it houses or it holds the DNA. And we know that the DNA contains our genetic information. Okay? So because of that, basically the cell nucleus is the control center of the cell. Okay? Now, the nucleus is actually surrounded by a covering called the nuclear envelope. Its location is it's found at the edge of the nucleus, or basically outside of the nucleus. And the nuclear envelope consists of an outer and an inner membrane. And between the outer and inner membrane is a narrow space between them. Now, your nuclear envelope and the nucleus actually has several holes, no? And these holes are called nuclear pores. Now, what's the function of those holes found on the nucleus and on the nuclear pore? The function of the pores are, this is where materials actually pass in and pass out of the nucleus. So this green structure that you see here, this represents your nuclear envelope. Again, it surrounds the entirety of the nucleus. And the nuclear pores are holes found on the surface of the nucleus and on the surface of the nuclear envelope. So as mentioned, the nucleus contains our DNA. And each human cell contains 23 pairs of chromosomes. What are chromosomes? These are your chromosomes. So chromosomes are tightly coiled. They are tightly coiled or very condensed arrangement of your chromatin. What is chromatin naman? Chromatin is the loose arrangement of your proteins and your DNA. So sa madaling sabi, yung DNA mo, may binabalot siya na proteins. And then, yung combination of DNA and protein, kapag sila ay hindi pa tightly coiled or hindi pa ganito ka-compact, they are called chromatin. But once they become very condensed and very compact, it will then be called a chromosome. And a human cell usually contains 23 pairs of chromosomes. There are 23 pairs of chromosomes. All right? So, when does your chromatin become a chromosome? When cell division is about to happen. So, as said in the bullet, when a cell prepares to divide, the chromosomes become tightly coiled and they become visible under the microscope. Okay? Now, at the very center of the nucleus, there is a prominent part as well, and it's called the nucleolus. The nucleolus are diffuse bodies with no surrounding membrane. So, hindi siya katulad ni nucleus na may nuclear envelope. Si nucleolus, wala siyang covering. And in some cells, there is only one nucleolus, but in other nucleus, there are several nucleoli. Okay, so iba-iba. May mga cells na isa lang ang nucleolus inside the nucleus. May mga cells din na several nucleolus in a single nucleus. Okay? So anong function ng nucleolus natin? Or remember this, they produce ribosomes. They produce ribosomes. And these ribosomes, later on, will know what its function is. These ribosomes exit the nucleus and the nuclear envelope through the nuclear pores. So, si nucleolus, it's found at the very center of the nucleus, di ba? So, makikreate siya. It will create a lot of ribosomes. And these ribosomes will exit the nucleus through the nuclear pores. So, putting everything together, this is your nucleus. The nucleus has an outer covering called the nuclear envelope. And the nuclear envelope and the nucleus has several holes surrounding it. And those holes are called nuclear pore. Inside the nucleus, we have the nucleolus. Some cells have one, some cells have a lot of nucleolus. And the function of the nucleolus is that it produces ribosomes. And these ribosomes would exit the nucleus and the nuclear envelope through the nuclear pore. Now, let's go to the ribosomes. What are ribosomes? Again, ribosomes are produced in the nucleolus, and the function of the ribosome is to produce proteins. Okay? So, it produces proteins. And these ribosomes, they would commonly attach to the rough endoplasmic reticulum, or they can float freely in the cytoplasm. Okay? After ma-produce ng ribosomes, after they exit the nucleus and the nuclear pore, they can either attach to the rough endoplasmic reticulum or they can float freely in the cytoplasm. And if they float freely in the cytoplasm, they are called your free ribosomes. Alright? They are called your free ribosomes. So as you can see here, these blue dots that you can see inside are actually your ribosomes. And they would exit the nuclear pore, and once outside, they can either attach themselves to the rough endoplasmic reticulum, such as this one, or they can float freely in the cytoplasm and be called your free ribosomes. Next, let's talk about the endoplasmic reticulum. And there are two types, your rough and smooth endoplasmic reticulum. So, ang endoplasmic reticulum natin, they extend from the outer nuclear membrane towards the cytoplasm. So basically, your endoplasmic reticulum is connected to your nuclear envelope. Okay? That's why look at this. It begins after the nuclear envelope and extends towards the cytoplasm. Okay? So dun siya nagsisimula. After nuclear envelope and nuclear pore, start na ng endoplasmic reticulum mo. And there are two types, your rough and smooth endoplasmic reticulum. Another description that we see here is that they are formed from sacs and tubules. So when we say sacs, these are the flattened appearance of the endoplasmic reticulum. So sila yung mga parang flat na maze looking. And then when we say tubules, ito yan. Para silang mga tubes, no? And just by looking at it, you'll be able to determine that this flat endoplasmic reticulum with many, many dots is your rough endoplasmic reticulum. And do you know what these blue structures are? Ano yung naka-attach sa rough endoplasmic reticulum? What's attached to it? Your ribosomes. So, what makes the RER or the rough endoplasmic reticulum rough looking are the attached ribosomes. And the smooth endoplasmic reticulum is called smooth endoplasmic reticulum because, apologies for that. So they are called smooth endoplasmic reticulum because they do not have any ribosomes attached to them. All right? Yeah. So let's single in on the rough endoplasmic reticulum. Again, they are found in the cytoplasm right after the nuclear envelope. And their characteristic is they appear rough looking because of the ribosomes attached to them. Now, what is the function of the RER? This is a site for protein synthesis. Again, what produces your proteins are your ribosomes. But the place where they are produced is the rough endoplasmic reticulum. I hope that's clear. Next is the smooth endoplasmic reticulum. Again, located in the cytoplasm, as you can see here. Usually after the rough endoplasmic reticulum, that's where you will find your smooth endoplasmic reticulum. They are described as tubular and smooth appearing because they have no ribosomes. They function naman for lipid synthesis. So they produce your cholesterol and other lipids. Okay? Moreover, they also function for detoxification. So, the smooth endoplasmic reticulum are the ones that clean the cell. They detoxify, remove the toxins of the cells. Okay? So, what organ in the body actually removes our toxins? It's our liver. So, basically, the liver is able to detoxify the body because it has a lot of smooth endoplasmic reticulum. Please do remember that. Might come out in the exam. Your liver has a lot of SER. And the last function of your smooth endoplasmic reticulum, it stores calcium in your skeletal muscles. And you will eventually find out na important ang ating smooth endoplasmic reticulum for muscle contraction. So it keeps your calciums. Okay? Next, we go to the Golgi Apparatus, which in other books, they would call it the Golgi Complex. So the Golgi Complex is a series of closely packed, curved, and membrane-bound sacs. So we saw the word sacs already a while ago, di ba? It's similar to the appearance of your rough endoplasmic reticulum, except that it's curved. Unlike your rough endoplasmic reticulum kanina, medyo there are corners to it, no? So your Golgi apparatus is found on the cytoplasm. And let's take a look first. Ayan. So commonly, after siya ng rough and smooth endoplasmic reticulum, you'll find this flat, curved, membranous sacs like that, no? And what's their function? They collect, they modify, they package, and distribute the proteins and lipids that were synthesized at your endoplasmic reticulum. In other words, the Golgi apparatus, it purifies, it packages, it makes sure that all the proteins and lipids are functioning well. Yun ang trabaho niya. Para siyang quality assurance. Okay? And after niya ma-make sure that your proteins and lipids are of good quality, then it will form vesicles. Specifically, secretory vesicles. No? And dun niya ilalagay yung proteins and lipids inside your secretory vesicles. And we've actually encountered secretory vesicles already in the first video. Diba? We use secretory vesicles for exocytosis. Remember, when we want to bring substances towards the outer or the exterior of the cell, we put them in secretory vesicles. So secretory vesicles distributes this proteins, lipids, and etc. out of the cell. So let's take a look. Again, the green highlighted part are your Golgi apparatus and the circular substances that actually bud off from your Golgi apparatus. When I say bud off, humihiwalay sila mula doon sa Golgi apparatus. Okay? Humihiwalay sila and they fuse with the cell membrane by exocytosis to release those substances. Okay? Under the electron microscope, this is how it would look like. And those circles that you see outside, those are secretory vesicles. Next, we have your lysosomes. Your lysosomes are also created by your Golgi apparatus. As you can see here, it forms vesicles, lysosomes, and other vesicles. So lysosomes are created by your Golgi apparatus. Alrighty? Now, unlike your secretory vesicles na ang laman ay protein and lipids, your lysosomes contain enzymes. Okay, they contain enzymes and these enzymes function as intracellular digestive systems. Meaning, di ba may endocytosis, and endocytosis is the process of getting substances and bringing them inside the cell. So, we have cell eating and cell drinking. So, basically, the cell eats and drinks. So, itong mga food or substances that the cell eats and drinks, ang nagdadigest sa kanila are your lysosomes. So, the vesicles, after endocytosis, will fuse with the lysosomes to break down these materials found in the endocytotic vesicles. Okay? So now, we are putting the concepts together. So this is how it would look like. Again, you have substances outside the cell and they will be brought in through endocytosis. So pwedeng solid, pwedeng liquid, depende na lang. So either phagocytosis or pinocytosis. Then they will merge with your lysosomes created by your Golgi apparatus. They will merge and the enzymes found inside will digest the substances. Okay? Yan. Next, we have your peroxisomes. Peroxisomes are also found in the cytoplasm. And peroxisomes, they are membrane-bound vesicles that contain enzymes that actually break down your fatty acids, your amino acids, and hydrogen peroxide. So, ang laman niya is enzyme that destroys fatty acids, amino acids, and hydrogen peroxide. So, hydrogen peroxide kasi is a byproduct of fatty acid and amino acid breakdown. And hydrogen peroxide can be toxic to a cell. Kaya dapat masira yung hydrogen peroxide. So the enzymes in the peroxisomes will break down the hydrogen of hydrogen peroxide. Okay? Next, something that is very familiar to everybody, your mitochondria, also known as the powerhouse of the cell. So they're located again in the cytoplasm, and your mitochondria has an inner and an outer membrane separated by a space. So let me show you. There you go. We have an outer membrane and an inner membrane, as you can see here. Now let's try to give them some names. The outer membrane has a smooth contour, but the inner membrane has numerous folds called cristae. So the folds of the inner membrane is called cristae, and they usually project into the interior, meaning yung folds ay nakaturo paloob sa gitna ng cell. Nagpo-fold sila towards the middle of the cell. And that's the meaning of the infolding of the inner membrane. Okay? So that's what we call cristae. Sorry. Now, the spaces in between the cristae is called matrix. Okay? So the material or the spaces in between each cristae are called matrix. And inside the matrix, you will find enzymes and also a mitochondrial DNA. So pag tinanong kayo sa exam, which two organelles contain DNA? Sagot, nucleus and mitochondria. Alrighty? Yeah. So why is it called the powerhouse of the cell? Because it is responsible for producing ATP. And what is the process of ATP production in the mitochondria? It's called aerobic metabolism. Do not forget that. It will be asked. It's called aerobic metabolism. Why aerobic? Because it uses oxygen. Okay, it uses oxygen to produce ATP. So commonly, the cells in the body that needs large energy requirements usually have more mitochondria than the cells that do not require energy. So anong cells kaya in our body needs a lot of mitochondria? Muscle cells, brain cells, 'di ba? Kasi kailangan nila ng energy. So this is how it would look like. Again, this is your mitochondria. It has an outer membrane, a smooth contour, and then it has an inner membrane. And the outer and the inner membrane is separated by a space. Alrighty? Now, the space naman in between your cristae, or the folds, are called matrix, and they contain enzymes and DNA. So this is how it would look like. Here you see the cristae, and the spaces between these green structures are called matrix. We're nearing the ends of our cell anatomy and physiology. Next, we have the cytoskeleton. So ang cytoskeleton is found both in the cytoplasm and in the cell membrane. So what's the role of the cytoskeleton? It gives the internal framework to the cell. Kumbaga para siyang skeleton ng body natin, di ba? The skeletal system, yung mga buto natin, it gives the framework, the general shape, di ba, of the body. Ganon din yung cytoskeleton. It gives the general framework or shape of the cell. Now, this cytoskeleton are made up of proteins. Okay? They are made up of proteins, and they hold the organelles in place. They enable the cell to also change in shape. We'll go into that in a little while. And there are three types of cytoskeleton: your microtubules, your microfilaments, and your intermediate filaments. So let me show you the three first. Here we go. Again, you find them on the cell membrane, and you find them in the cytoplasm. Yeah, there are three types: filaments, tubules, intermediate filaments. So, anong number one difference nila? Diameter. Pinakamalaki, microtubule. Sumunod, intermediate. That's why they are called intermediate. They are the middle. And then, the thinnest is the microfilament. Okay? So, let's start with the microtubules. Microtubules, again, is the largest of the three. And they are characterized as hollow structures or hollow tubes made up of proteins. Okay? Now, what is the major function of microtubules? Number one, they support the cytoplasm of the cells. Kaya nga here in this picture, you can see several big tubes in the cytoplasm. So they support the general shape of the cytoplasm. Secondly, microtubules are important for cell division. So later, in the third video, we will talk about cell division. And then, lastly, microtubules are important components of your cilia and flagella, which we will discuss in a little while. Alrighty? As you can see here also, you find a lot of microtubules surrounding the cytoplasm. And even here, you see it, no? It's very distributed all over. It gives the general framework or general shape of the cytoplasm. Next is the thinnest one, the microfilament. So they are small fibers made up of proteins, and the main function is to determine cell shape. They give shape to the cell, microfilaments. Okay? Moreover, they are important in the shortening or contraction of your cells. Kaya nga shape, 'di ba? nababago-bago nila yung shape ng cells mo. So, when we discuss the muscular system, you will find out how important your microfilaments are. Lastly, of the three cytoskeleton, we have the intermediate filament. Kaya nga siya, it's called intermediate because it's the medium diameter. So, they are smaller than your microtubules but bigger than your microfilaments. And what is their main function? Mechanical support. Mechanical support. In fact, there are so much intermediate filaments in our skin, specifically sa keratin. Okay? Keratin. So this is a summary picture of the three. Now, just a few more organelles. Next, we have centrioles. Ang centrioles naman, they are arranged in triplets of microtubules. So, gawa sila sa microtubules na naka-arrange in triplets in groups of 9. So, there should be a number 9 here. In groups of 9. So, let me show you. Ayan, this is one centriole. So, we have 9. Let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9. So, 9 triplets. As you can see, each has 3 microtubules. Yan. Those are your centrioles. So, ang centrioles natin, they are important for cell division. Okay? They are important in the process of mitosis or cell division. Next, we have your cilia and your flagella. Again, they are made up of your microtubules. Diba? So, cilia, there are often many cilia found in each cell and they are commonly found on the surface of the cells. Pag sinabing surface, sa labas lang sila ng cell nakikita. So, they are usually cylindrical structures para silang mga galamay. No? They are responsible for the movement of materials over the top of the cell, such as your mucus. So, para silang mga galamay that moves substances pag pumapatong sa cell. So, can you just imagine? They are like seaweeds on the seafloor. They are continually moving. They are like, parang mga seaweeds sila na nagmo-move. So that lahat ng substances na pumapatong sa kanila or dumidikit sa kanila, natatanggal nila. Okay? That's your cilia. Next is your flagella. Again, still made up of your microtubules. But flagella compared to cilia are much longer. Mas mahaba ang flagella. And kung ang cilia marami, many per cell, in flagella, they only have one. One flagella per cell. So, one good example of a cell with flagella is your sperm cells. So, ito yung flagella, itong parang extension sa ilalim. Now, what's the function of a flagella? It helps propel or move the cell. So, it acts as if like it's a tail, that nagwawaggle yung tail na yun, no? In order to move the cell from one place to another. Lastly, we have the microvilli. Microvilli are extensions of the cell membrane and they are made up of microfilaments. Okay? They do not move unlike your cilia and flagella. Diba sa cilia, sabi ko, para silang seaweeds under on the floor of the sea. So, nagmo-move. But your microvilli does not move. Okay, they are not for movement. So there are numerous microvilli on the cells. And what is the function? To increase the surface area. Why? Because increased surface area is important for absorption. So let me show you. Ayan. So itong cell na to, it has an area where there is microvilli and a lot of area without microvilli. So itong buong to, that's the surface area, right? So, pag may gusto siyang i-absorb, gagamitin niya yung surface niya to absorb it. Sorry. Yan. Now, for important substances that needs extra absorption, dapat may microvilli. Bakit? Kasi pag straight lang siya, dito lang siya ma-absorb. No? Dito lang siya ma-absorb. Pero pag may microvilli ka, dito hanggang sa mga gilid niya, kaya niya mag-absorb. Yan. So, up and down, kaya nila mag-absorb. So, imagine mo, Kung straight lang yan lang yung may capability mag-absorb. But pag may microvilli, kahit yung mga gilid niya, no, tumataas, tumataas yung ability niya to absorb, 'di ba? So it increases the surface it increases the surface area for absorption. So that's the last organelle I want to talk about in your cell anatomy and physiology. There is a third video for your cell activity. So hopefully you learned something. See you in the next video.